Animal welfare. Since the 18th century, people have become increasingly concerned about the welfare of farm animals. Possible measures of welfare include longevity, behavior, physiology, reproduction, freedom from disease, and freedom from immunosuppression. Standards and laws for animal welfare have been created worldwide, broadly in line with the most widely held position in the Western world, a form of utilitarianism, that it is morally acceptable for humans to use non-human animals, provided that no unnecessary suffering is caused, and that the benefits to humans outweigh the costs to the livestock. An opposing view is that animals have rights, should not be regarded as property, or not necessary to use, and should never be used by humans. 95, 96, 97, 98, 99, live export of animals has risen to meet increased global demand for livestock such as in the Middle East. Animal rights activists have objected to long-distance transport of animals. One result was the banning of live exports from New Zealand in 2003. 100. In culture. Opening of the budget, or, John Bull giving his breeches to save his bacon, Note 1 by James Gilray d. 1815. Since the 18th century, the farmer John Bull has represented English national identity, first in John Arbuthnot's political satires, and soon afterwards in cartoons by James Gilray and others including John Tenniel. He likes food, beer, dogs, horses, and country sports. He is practical and down-to-earth, and anti-intellectual. 101. Farm animals are widespread in books and songs for children. The reality of animal husbandry is often distorted, softened, or idealized, giving children an almost entirely fictitious account of farm life. The books often depict happy animals free to roam in attractive countryside, a picture completely at odds with the realities of the impersonal, mechanized activities involved in modern intensive farming. 102. Dressed pigs in Beatrix Potter's 1913 The Tale of Pigling Bland. Pigs, for example, appear in several of Beatrix Potter's little books, as Piglet in A. A. Milne's Winnie the Pooh stories, and somewhat more darkly with a hint of animals going to slaughter, as Babe in Dick King Smith's The Sheep Pig, and as Wilbur in E. B. White's Charlotte's Web. 103. Pigs tend to be bearers of cheerfulness, good humor and innocence. Many of these books are completely anthropomorphic, dressing farm animals in clothes and having them walk on two legs, live in houses, and perform human activities. 102. The Children's Song. Old MacDonald had a farm, describes a farmer named MacDonald and the various animals he keeps, celebrating the noises they each make. 104. Many urban children experience animal husbandry for the first time at a petting farm. In Britain, some 5 million people a year visit a farm of some kind. This presents some risk of infection, especially if children handle animals and then fail to wash their hands. A strain of E. coli infected 93 people who had visited a British interactive farm in an outbreak in 2009. 105. Historic farms such as those in the United States offer farm stays in a carefully curated version of farming to those willing to pay for it. 106. Sometimes giving visitors a romantic-ized image of a pastoral idyll from an unspecified time in the pre-industrial past. 106. See also. Marker-assisted selection. Marker-assisted selection or marker-aided selection (MAS) is an indirect selection process where a trait of interest is selected based on a marker morphological, biochemical or DNA, RNA variation, linked to a trait of interest e.g. productivity, disease resistance, abiotic stress tolerance, and quality rather than on the trait itself. 1, 2, 3, 4, 5. This process has been extensively researched and proposed for plant and animal breeding. 5. For example, using MAS to select individuals with disease resistance involves identifying a marker allele that is linked with disease resistance rather than the level of disease resistance. The assumption is that the marker associates at high frequency with the gene or quantitative trait locus (QTL) of interest, due to genetic linkage close proximity, on the chromosome, of the marker locus and the disease resistance determining locus. MAS can be useful to select for traits that are difficult or expensive to measure, exhibit low heritability and or are expressed late in development. At certain points in the breeding process the specimens are examined to ensure that they express the desired trait. Marker types. The majority of MAS work in the present era uses DNA-based markers. 5. However, the first markers that allowed indirect selection of a trait of interest were morphological markers. In 1923, Carl Sachs first reported association of a simply inherited genetic marker with a quantitative trait in plants when he observed segregation of seed size associated with segregation for a seed coat color marker in beans, Phaseolus vulgaris L. 6. In 1935, J. Rasmussen demonstrated linkage of flowering time, a quantitative trait, in peas with a simply inherited gene for flower color. 7. Markers may be morphological. These were the first markers loci available that have an obvious impact on the morphology of plants. These markers are often detectable by eye, by simple visual inspection. Examples of this type of marker include the presence or absence of an on, leaf sheath coloration, height, grain color, aroma of rice, etc. In well-characterized crops like maize, tomato, pea, barley or wheat, tens or hundreds of genes that determine morphological traits have been mapped to specific chromosome locations. Biochemical, a protein that can be extracted and observed, for example, isozymes and storage proteins. Cytological markers are chromosomal features that can be identified through microscopy. These generally take the form of chromosome bands, regions of chromatin that become impregnated with specific dyes used in cytology. The presence or absence of a chromosome band can be correlated with a particular trait, indicating that the locus responsible for the trait is located within or near, tightly linked to the banded region. Morphological and cytological markers form the backbone of early genetic studies in crops such as wheat and maize. 8. DNA-based, including microsatellites also known as short tandem repeats, STRs, or simple sequence repeats, SSRs, restriction fragment length polymorphism, RFLP, random amplification of polymorphic DNA, RAPD, amplified fragment length polymorphism, AFLP, and single nucleotide polymorphisms, SNPs. 9. Positive and negative selectable markers. The following terms are generally less relevant to discussions of moss in plant and animal breeding, but are highly relevant in molecular biology research. Positive selectable markers are selectable markers that confer selective advantage to the host organism. 10. An example would be antibiotic resistance, which allows the host organism to survive antibiotic selection. Negative selectable markers are selectable markers that eliminate or inhibit growth of the host organism upon selection. 11. An example would be thymidine kinase, which makes the host sensitive to ganciclovir selection. A distinction can be made between selectable markers, which eliminate certain genotypes from the population, and screenable markers, which cause certain genotypes to be readily identifiable, at which point the experimenter must score or evaluate the population and act to retain the preferred genotypes. Most moss uses screenable markers rather than selectable markers. Gene versus marker. The gene of interest directly causes production of proteins or RNA that produce a desired trait or phenotype, whereas markers, a DNA sequence or the morphological or biochemical markers produced due to that DNA, are genetically linked to the gene of interest. The gene of interest and the marker tend to move together during segregation of gametes due to their proximity on the same chromosome and concomitant reduction and recombination chromosome crossover events between the marker and gene of interest. For some traits, the gene of interest has been discovered and the presence of desirable alleles can be directly assayed with a high level of confidence. However, if the gene of interest is not known, markers linked to the gene of interest can still be used to select for individuals with desirable alleles of the gene of interest. When markers are used, there may be some inaccurate results due to inaccurate tests for the marker. There also can be false positive results when markers are used, due to recombination between the marker of interest and gene, or QTL. A perfect marker would elicit no false positive results. The term perfect marker is sometimes used when tests are performed to detect a SNP or other DNA polymorphism in the gene of interest, if that SNP or other polymorphism is the direct cause of the trait of interest. The term marker is still appropriate to use when directly assaying the gene of interest, because the test of genotype is an indirect test of the trait or phenotype of interest. Important properties of ideal markers for moss. An ideal marker has easy recognition of phenotypes, ideally all possible phenotypes, homo and heterozygotes, from all possible alleles. Demonstrates measurable differences in expression between trait types or gene of interest alleles, early in the development of the organism. Testing for the marker does not have variable success depending on the allele at the marker locus or the allele at the target locus, the gene of interest that determines the trait of interest. Low or no interaction among the markers, allowing the use of many at the same time in a segregating population. Abundant in number. Polymorphic. Drawbacks of morphological markers. Morphological markers are associated with several general deficits that reduce their usefulness, including the delay of marker expression until late into the development of the organism, allowing dominance to mask the underlying genetics. Pleiotropy, which does not allow easy and parsimonious inferences to be drawn from one gene to one trait. Confounding effects of genes unrelated to the gene or trait of interest but which also affect the morphological marker, epistasis. Frequent confounding effects of environmental factors which affect the morphological characteristics of the organism. To avoid problems specific to morphological markers, DNA-based markers have been developed. They are highly polymorphic, exhibit simple inheritance, often codominant, are abundant throughout the genome, are easy and fast to detect, exhibit minimum pleiotropic effects, and detection is not dependent on the developmental stage of the organism. Numerous markers have been mapped to different chromosomes in several crops including rice, wheat, maize, soybean and several others, and in livestock such as cattle, pigs and chickens. Those markers have been used in diversity analysis, parentage detection, DNA fingerprinting, and prediction of hybrid performance. Molecular markers are useful in indirect selection processes, enabling manual selection of individuals for further propagation. Selection for major genes linked to markers. Major genes that are responsible for economically important characteristics are frequent in the plant kingdom. Such characteristics include disease resistance, male sterility, 12th self-incompatibility, and others related to shape, color, and architecture of whole plants and are often of mono or oligogenic in nature. The marker loci that are tightly linked to major genes can be used for selection and are sometimes more efficient than direct selection for the target gene. Such advantages in efficiency may be due, for example, to higher expression of the marker mRNA in such cases that the marker is itself a gene. Alternatively, in such cases that the target gene of interest differs between two alleles via difficult-to-detect single nucleotide polymorphism, an external marker, be it another gene or a polymorphism that is easier to detect, such as a short tandem repeat, may present as the most realistic option. Situations that are favorable for molecular marker selection. There are several indications for the use of molecular markers in the selection of a genetic trait. Situations such as the selected character is expressed late in plant development, like fruit and flower features or adult characters with a juvenile period, so that it is not necessary to wait for the organism to become fully developed before arrangements can be made for propagation. The expression of the target gene is recessive, so that individuals which are heterozygous positive for the recessive allele can be crossed to produce some homozygous offspring with the desired trait. There are special conditions for expression of the target genes, as in the case of breeding for disease and pest resistance, where inoculation with the disease or subjection to pests would otherwise be required. Sometimes inoculation methods are unreliable and sometimes field inoculation with the pathogen is not even allowed for safety reasons. Moreover, sometimes expression is dependent on environmental conditions. The phenotype is affected by two or more unlinked genes, epistatus. For example, selection for multiple genes which provide resistance against diseases or insect pests for gene pyramiding. The cost of genotyping, for example, the molecular marker assays needed here, is decreasing, thus increasing the attractiveness of MOS as the development of the technology continues. Additionally, the cost of phenotyping performed by a human is a labor burden, which is higher in a developed country and increasing in a developing country. Steps for MOS. Generally, the first step is to map the gene or quantitative trait locus, QTL, of interest first by using different techniques and then using this information for marker-assisted selection. Generally, the markers to be used should be close to gene of interest, less than 5 recombination unit or cm, in order to ensure that only minor fraction of the selected individuals will be recombinants. Generally, not only a single marker but rather two markers are used in order to reduce the chances of an error due to homologous recombination. For example, if two flanking markers are used at same time with an interval between them of approximately 20 cm, there is higher probability, 99%, for recovery of the target gene. QTL mapping techniques. In plants, QTL mapping is generally achieved using biparental cross populations, a cross between two parents which have a contrasting phenotype for the trait of interest are developed. Commonly used populations are near isogenic lines, nils, recombinant inbred lines, RILs, doubled haploids, DH, back cross and F2. Linkage between the phenotype and markers which have already been mapped is tested in these populations in order to determine the position of the QTL. Such techniques are based on linkage and are therefore referred to as linkage mapping. A. Single-step MOS and QTL mapping. In contrast to two-step QTL mapping in MOS, a single-step method for breeding typical plant populations has been developed. 13-14. In such an approach, in the first few breeding cycles, markers linked to the trait of interest are identified by QTL mapping and later the same information is used in the same population. In this approach, pedigree structure is created from families that are created by crossing number of parents in 3-way or 4-way crosses. Both phenotyping and genotyping is done using molecular markers map the possible location of QTL of interest. This will identify markers and their favorable alleles. Once these favorable marker alleles are identified, the frequency of such alleles will be increased in response to marker-assisted selection is estimated. Marker alleles with desirable effect will be further used in next selection cycle or other experiments. High-throughput genotyping techniques. Recently, high-throughput genotyping techniques are developed which allows marker-aided screening of many genotypes. This will help breeders in shifting traditional breeding to marker-aided selection. One example of such automation is using DNA isolation robots, capillary electrophoresis and pipetting robots. One recent example of capillary system is Applied Biosystems 3130 genetic analyzer. This is the latest generation of 4 capillary electrophoresis instruments for the low to medium throughput laboratories. High throughput MOS is needed for crop breeding because current techniques are not cost-effective. Arrays have been developed for rice by Massola et al. 2009, wheat by Berard et al. 2009, Bernardo et al. 2015, and Rashid et al. 2016, legumes by Varshni et al. 2016, and various other crops, but all of these have also problems with customization, cost, flexibility, and equipment costs. 15. Use of MOS for back-cross breeding. A minimum of 5 or 6 back-cross generations are required to transfer a gene of interest from a donor, may not be adapted, to a recipient, recurrent, adapted cultivar. The recovery of the recurrent genotype can be accelerated with the use of molecular markers. If the F1 is heterozygous for the marker locus, individuals with the recurrent parent alleles at the marker locus in first or subsequent back-cross generations will also carry a chromosome tagged by the marker. Marker-assisted gene pyramiding. Gene pyramiding has been proposed and applied to enhance resistance to disease in insects by selecting for 2 or more than 2 genes at a time. For example, in rice such pyramids have been developed against bacterial blight and blast. The advantage of use of markers in this case allows to select for QTL allele-linked markers that have same phenotypic effect. MAS has also been proved useful for livestock improvement. 16. A coordinated effort to implement wheat durum, Triticum turgidum, and common wheat Triticum aestivum, marker-assisted selection in the U.S. as well as a resource for marker-assisted selection exists at the WheatCap Coordinated Agricultural Project website. See also. Association mapping. Genetically modified organism. A genetically modified organism, GMO, is any organism whose genetic material has been altered using genetic engineering techniques. The exact definition of a genetically modified organism and what constitutes genetic engineering varies, with the most common being an organism altered in a way that does not occur naturally by mating and/or natural recombination. 1. A wide variety of organisms have been genetically modified, GM, including animals, plants, and microorganisms. Genetic modification can include the introduction of new genes or enhancing, altering, or knocking out endogenous genes. In some genetic modifications, genes are transferred within the same species, across species creating transgenic organisms, and even across kingdoms. Creating a genetically modified organism is a multi-step process. Genetic engineers must isolate the gene they wish to insert into the host organism and combine it with other genetic elements, including a promoter and terminator region and often a selectable marker. A number of techniques are available for inserting the isolated gene into the host genome. Recent advancements using genome editing techniques, notably CRISPR, have made the production of GMOs much simpler. Herbert Boyer and Stanley Cohen made the first genetically modified organism in 1973, a bacterium resistant to the antibiotic kanamycin. The first genetically modified animal, a mouse, was created in 1974 by Rudolf Janisch, and the first plant was produced in 1983. In 1994, the Flavor Saver tomato was released, the first commercialized genetically modified food. The first genetically modified animal to be commercialized was the glowfish, 2003, and the first genetically modified animal to be approved for food use was the Aku Advantage salmon in 2015. Bacteria are the easiest organisms to engineer and have been used for research, food production, industrial protein purification, including drugs, agriculture, and art. There is potential to use them for environmental purposes or as medicine. Fungi have been engineered with much the same goals. Viruses play an important role as vectors for inserting genetic information into other organisms. This use is especially relevant to human gene therapy. There are proposals to remove the virulent genes from viruses to create vaccines. Plants have been engineered for scientific research, to create new colors in plants, deliver vaccines, and to create enhanced crops. Genetically modified crops are publicly the most controversial GMOs, in spite of having the most human health and environmental benefits. 2. Animals are generally much harder to transform and the vast majority are still at the research stage. Mammals are the best model organisms for humans. Livestock is modified with the intention of improving economically important traits such as growth rate, quality of meat, milk composition, disease resistance, and survival. Genetically modified fish are used for scientific research, as pets, and as a food source. Genetic engineering has been proposed as a way to control mosquitoes, a vector for many deadly diseases. Although human gene therapy is still relatively new, it has been used to treat genetic disorders such as severe combined immunodeficiency and Leber's congenital amaurosis. Many objections have been raised over the development of GMOs, particularly their commercialization. Many of these involve GM crops and whether food produced from them is safe and what impact growing them will have on the environment. Other concerns are the objectivity and rigor of regulatory authorities, contamination of non-genetically modified food, control of the food supply, patenting of life, and the use of intellectual property rights. Although there is a scientific consensus that currently available food derived from GM crops poses no greater risk to human health than conventional food, GM food safety is a leading issue with critics. Gene flow, impact on non-target organisms, and escape are the major environmental concerns. Countries have adopted regulatory measures to deal with these concerns. There are differences in the regulation for the release of GMOs between countries, with some of the most marked differences occurring between the US and Europe. Key issues concerning regulators include whether GM food should be labeled and the status of gene-edited organisms. Definition. The definition of a genetically modified organism, GMO, is not clear and varies widely between countries, international bodies, and other communities. At its broadest, the definition of a GMO can include anything that has had its genes altered, including by nature. 3.4 Taking a less broad view, it can encompass every organism that has had its genes altered by humans, which would include all crops and livestock. In 1993, the Encyclopedia Britannica defined genetic engineering as any of a wide range of techniques. Among them artificial insemination, in vitro fertilization e.g., test tube, babies, sperm banks, cloning, and gene manipulation. 5. The European Union (EU) included a similarly broad definition in early reviews, specifically mentioning GMOs being produced by selective breeding and other means of artificial selection. 6. These definitions were promptly adjusted with a number of exceptions added as a result of pressure from scientific and farming communities, as well as developments in science. The EU definition later excluded traditional breeding, in vitro fertilization, induction of polyploidy, mutation breeding, and cell fusion techniques that do not use recombinant nucleic acids or a genetically modified organism in the process. 7, 8, 9 Another approach was the definition provided by the Food and Agriculture Organization, the World Health Organization, and the European Commission, stating that the organisms must be altered in a way that does not occur naturally by mating and/or natural recombination. 10, 11, 12 Progress in science, such as the discovery of horizontal gene transfer being a relatively common natural phenomenon, further added to the confusion on what occurs naturally, which led to further adjustments and exceptions. 13 There are examples of crops that fit this definition, but are not normally considered GMOs. 14 For example, the grain crop triticale was fully developed in a laboratory in 1930 using various techniques to alter its genome. 15 Genetically engineered organism, GEO, can be considered a more precise term compared to GMO when describing organisms' genomes that have been directly manipulated with biotechnology. 16. 8. The Cartagena Protocol on Biosafety used the synonym living modified organism, LMO, in 2000 and defined it as any living organism that possesses a novel combination of genetic material obtained through the use of modern biotechnology. 17. Modern biotechnology is further defined as in vitro nucleic acid techniques, including recombinant deoxyribonucleic acid, DNA, and direct injection of nucleic acid into cells or organelles, or fusion of cells beyond the taxonomic family. 18. Originally, the term GMO was not commonly used by scientists to describe genetically engineered organisms until after usage of GMO became common in popular media. 19. The United States Department of Agriculture (USDA) considers GMOs to be plants or animals with heritable changes introduced by genetic engineering or traditional methods, while GEO specifically refers to organisms with genes introduced, eliminated, or rearranged using molecular biology, particularly recombinant DNA techniques, such as transgenesis. 20. The definitions focus on the process more than the product, which means there could be GMOs and non-GMOs with very similar genotypes and phenotypes. 21, 22 This has led scientists to label it as a scientifically meaningless category. 23 Saying that it is impossible to group all the different types of GMOs under one common definition. 24 It has also caused issues for organic institutions and groups looking to ban GMOs. 25, 26 It also poses problems as new processes are developed. The current definitions came in before genome editing became popular and there is some confusion as to whether they are GMOs. The EU has adjudged that they're 27 changing their GMO definition to include organisms obtained by mutagenesis, but has excluded them from regulation based on their long safety record, and that they have conventionally been used in a number of applications. 9. In contrast, the USDA has ruled that gene-edited organisms are not considered GMOs. 28. Even greater inconsistency and confusion is associated with various non-GMO or GMO-free labeling schemes in food marketing, where even products such as water or salt, which do not contain any organic substances and genetic material and thus cannot be genetically modified by definition, are being labeled to create an impression of being more healthy. 29, 30, 31. Production. A gene gun uses biolistics to insert DNA into plant tissue. Creating a genetically modified organism, GMO, is a multi-step process. Genetic engineers must isolate the gene they wish to insert into the host organism. This gene can be taken from a cell. 32, or artificially synthesized. 33. If the chosen gene or the donor organism's genome has been well studied, it may already be accessible from a genetic library. The gene is then combined with other genetic elements, including a promoter and terminator region and a selectable marker. 34. A number of techniques are available for inserting the isolated gene into the host genome. Bacteria can be induced to take up foreign DNA, usually by exposed heat shock or electroporation. 35. DNA is generally inserted into animal cells using microinjection, where it can be injected through the cell's nuclear envelope directly into the nucleus, or through the use of viral vectors. 36. In plants, the DNA is often inserted using agrobacterium-mediated recombination. 37. 38. Biolistics. 39. Or electroporation. As only a single cell is transformed with genetic material, the organism must be regenerated from that single cell. In plants, this is accomplished through tissue culture. 40. 41. In animals, it is necessary to ensure that the inserted DNA is present in the embryonic stem cells. 37. Further testing using PCR, southern hybridization, and DNA sequencing is conducted to confirm that an organism contains the new gene. 42. Traditionally, the new genetic material was inserted randomly within the host genome. Gene targeting techniques, which creates double-stranded breaks and takes advantage on the cell's natural homologous recombination repair systems, have been developed to target insertion to exact locations. Genome editing uses artificially engineered nucleases that create breaks at specific points. There are 4 families of engineered nucleases: meganucleases, 43-44, zinc finger nucleases, 45-46, transcription activator-like effector nucleases, TALENs, 47-48, and the Cas9 guide RNA system, adapted from CRISPR. 49-50, TALEN and CRISPR are the 2 most commonly used, and each has its own advantages. 51, TALENs have greater target specificity, while CRISPR is easier to design and more efficient. 51. History. Herbert Boyer pictured and Stanley Cohen created the first genetically modified organism in 1973. Humans have domesticated plants and animals since around 12,000 BCE, using selective breeding or artificial selection, as contrasted with natural selection. Production. A gene gun uses biolistics to insert DNA into plant tissue. Creating a genetically modified organism, GMO, is a multi-step process. Genetic engineers must isolate the gene they wish to insert into the host organism. This gene can be taken from a cell. 32, or artificially synthesized. 33. If the chosen gene or the donor organism's genome has been well studied, it may already be accessible from a genetic library. The gene is then combined with other genetic elements, including a promoter and terminator region and a selectable marker. 34. A number of techniques are available for inserting the isolated gene into the host genome. Bacteria can be induced to take up foreign DNA, usually by exposed heat shock or electroporation. 35. DNA is generally inserted into animal cells using microinjection, where it can be injected through the cell's nuclear envelope directly into the nucleus, or through the use of viral vectors. 36. In plants, the DNA is often inserted using agrobacterium-mediated recombination. 37. 38. Biolistics. 39. Or electroporation. As only a single cell is transformed with genetic material, the organism must be regenerated from that single cell. In plants, this is accomplished through tissue culture. 40. 41. In animals, it is necessary to ensure that the inserted DNA is present in the embryonic stem cells. 37. Further testing using PCR, southern hybridization, and DNA sequencing is conducted to confirm that an organism contains the new gene. 42. Traditionally, the new genetic material was inserted randomly within the host genome. Gene targeting techniques, which creates double-stranded breaks and takes advantage on the cell's natural homologous recombination repair systems, have been developed to target insertion to exact locations. Genome editing uses artificially engineered nucleases that create breaks at specific points. There are 4 families of engineered nucleases: meganucleases, 43-44, zinc finger nucleases, 45-46, transcription activator-like effector nucleases, TALENs, 47-48, and the Cas9 guide RNA system, adapted from CRISPR. 49-50, TALEN and CRISPR are the 2 most commonly used, and each has its own advantages. 51, TALENs have greater target specificity, while CRISPR is easier to design and more efficient. 51. History. Herbert Boyer pictured and Stanley Cohen created the first genetically modified organism in 1973. Humans have domesticated plants and animals since around 12,000 BCE, using selective breeding or artificial selection, as contrasted with natural selection. 52-25 The process of selective breeding, in which organisms with desired traits, and thus with the desired genes, are used to breed the next generation and organisms lacking the trait are not bred, is a precursor to the modern concept of genetic modification. 53. 154. 1. Various advancements in genetics allowed humans to directly alter the DNA and therefore genes of organisms. In 1972, Paul Berg created the first recombinant DNA molecule when he combined DNA from a monkey virus with that of the lambda virus. 55. 56. Herbert Boyer and Stanley Cohen made the first genetically modified organism in 1973. 57. They took a gene from a bacterium that provided resistance to the antibiotic kanamycin, inserted it into a plasmid and then induced other bacteria to incorporate the plasmid. The bacteria that had successfully incorporated the plasmid was then able to survive in the presence of kanamycin. 58. Boyer and Cohen expressed other genes in bacteria. This included genes from the toad Xenopus laevis in 1974, creating the first GMO expressing a gene from an organism of a different kingdom. 59. In 1974, Rudolf Janisch created the first genetically modified animal. In 1974, Rudolf Janisch created a transgenic mouse by introducing foreign DNA into its embryo, making it the world's first transgenic animal. 60. 61. However, it took another 8 years before transgenic mice were developed that passed the transgene to their offspring. 62. 63. Genetically modified mice were created in 1984 that carried cloned oncogenes, predisposing them to developing cancer. 64. Mice with genes removed, termed a knockout mouse, were created in 1989. The first transgenic livestock were produced in 1985, 65, and the first animal to synthesize transgenic proteins in their milk were mice in 1987. 66. The mice were engineered to produce human tissue plasminogen activator, a protein involved in breaking down blood clots. 67. In 1983, the first genetically engineered plant was developed by Michael W. Bevan, Richard B. Flavel and Mary Del Chilton. They infected tobacco with Agrobacterium transformed with an antibiotic resistance gene and through tissue culture techniques were able to grow a new plant containing the resistance gene. 68. The gene gun was invented in 1987, allowing transformation of plants not susceptible to Agrobacterium infection. 69. In 2000, vitamin A enriched golden rice was the first plant developed with increased nutrient value. 70. In 1976, Genentech, the first genetic engineering company, was founded by Herbert Boyer and Robert Swanson. A year later, the company produced a human protein, somatostatin, in E. coli. Genentech announced the production of genetically engineered human insulin in 1978. 71. The insulin produced by bacteria, branded Humulin, was approved for release by the Food and Drug Administration in 1982. 72. In 1988, the first human antibodies were produced in plants. 73. In 1987, a strain of Pseudomonas syringae became the first genetically modified organism to be released into the environment. 74. When a strawberry and potato field in California were sprayed with it. 75. The first genetically modified crop, an antibiotic-resistant tobacco plant, was produced in 1982. 76. China was the first country to commercialize transgenic plants, introducing a virus-resistant tobacco in 1992. 77. In 1994, Calgene attained approval to commercially release the Flavor Saver tomato, the first genetically modified food. 78. Also in 1994, the European Union approved tobacco engineered to be resistant to the herbicide bromoxynil, making it the first genetically engineered crop commercialized in Europe. 79. An insect-resistant potato was approved for release in the U.S. in 1995, 80, and by 1996 approval had been granted to commercially grow 8 transgenic crops and 1 flower crop, carnation, in 6 countries plus the EU. 81. In 2010, scientists at the J. Craig Venter Institute announced that they had created the first synthetic bacterial genome. They named it Cynthia and it was the world's first synthetic life form. 82. 83. The first genetically modified animal to be commercialized was the glowfish, a zebrafish with a fluorescent gene added that allows it to glow in the dark under ultraviolet light. 84. It was released to the U.S. market in 2003. 85. In 2015, Aku Advantage salmon became the first genetically modified animal to be approved for food use. 86. Approval is for fish raised in Panama and sold in the U.S. 86. The salmon were transformed with a growth hormone regulating gene from a Pacific Chinook salmon and a promoter from an ocean trout enabling it to grow year-round instead of only during spring and summer. 87. Left. Bacteria transformed with PGLO under ambient light. Right. Bacteria transformed with PGLO visualized under ultraviolet light. Bacteria were the first organisms to be genetically modified in the laboratory, due to the relative ease of modifying their chromosomes. 88. This ease made them important tools for the creation of other GMOs. Genes and other genetic information from a wide range of organisms can be added to a plasmid and inserted into bacteria for storage and modification. Bacteria are cheap, easy to grow, clonal, multiply quickly and can be stored at -80 degrees Celsius almost indefinitely. Once a gene is isolated, it can be stored inside the bacteria, providing an unlimited supply for research. 89. A large number of custom plasmids make manipulating DNA extracted from bacteria relatively easy. 90. Their ease of use has made them great tools for scientists looking to study gene function and evolution. The simplest model organisms come from bacteria, with most of our early understanding of molecular biology coming from studying Escherichia coli. 91. Scientists can easily manipulate an bacteria. Combine genes within the bacteria to create novel or disrupted proteins and observe the effect this has on various molecular systems. Researchers have combined the genes from bacteria and archaea, leading to insights on how these two diverged in the past. 92. In the field of synthetic biology, they have been used to test various synthetic approaches, from synthesizing genomes to creating novel nucleotides. 93, 94, 95. Bacteria have been used in the production of food for a long time, and specific strains have been developed and selected for that work on an industrial scale. They can be used to produce enzymes, amino acids, flavorings, and other compounds used in food production. With the advent of genetic engineering, new genetic changes can easily be introduced into these bacteria. Most food-producing bacteria are lactic acid bacteria, and this is where the majority of research into genetically engineering food-producing bacteria has gone. The bacteria can be modified to operate more efficiently, reduce toxic byproduct production, increase output, create improved compounds, and remove unnecessary pathways. 96 food products from genetically modified bacteria include alpha amylase, which converts starch to simple sugars, chymosin, which clots milk protein for cheese making, and pectinesterase, which improves fruit juice clarity. 97. The majority are produced in the US, and even though regulations are in place to allow production in Europe, as of 2015 no food products derived from bacteria are currently available there. 98. Genetically modified bacteria are used to produce large amounts of proteins for industrial use. The bacteria are generally grown to a large volume before the gene encoding the protein is activated. The bacteria are then harvested and the desired protein purified from them. 99. The high cost of extraction and purification has meant that only high-value products have been produced at an industrial scale. 100. The majority of these products are human proteins for use in medicine. 101. Many of these proteins are impossible or difficult to obtain via natural methods, and they are less likely to be contaminated with pathogens, making them safer. 99. The first medicinal use of GM bacteria was to produce the protein insulin to treat diabetes. 102. Other medicines produced include clotting factors to treat hemophilia, 103. Human growth hormone to treat various forms of dwarfism, 104. 105. Interferon to treat some cancers, erythropoietin for anemic patients, and tissue plasminogen activator which dissolves blood clots. 99. Outside of medicine they have been used to produce biofuels. 106. There is interest in developing an extracellular expression system within the bacteria to reduce costs and make the production of more products economical. 100. With a greater understanding of the role that the microbiome plays in human health, there is a potential to treat diseases by genetically altering the bacteria to, themselves, be therapeutic agents. Ideas include altering gut bacteria so they destroy harmful bacteria, or using bacteria to replace or increase deficient enzymes or proteins. One research focus is to modify lactobacillus, bacteria that naturally provide some protection against HIV, with genes that will further enhance this protection. If the bacteria do not form colonies inside the patient, the person must repeatedly ingest the modified bacteria in order to get the required doses. Enabling the bacteria to form a colony could provide a more long-term solution, but could also raise safety concerns as interactions between bacteria and the human body are less well understood than with traditional drugs. There are concerns that horizontal gene transfer to other bacteria could have unknown effects. As of 2018 there are clinical trials underway testing the efficacy and safety of these treatments. 107. For over a century, bacteria have been used in agriculture. Crops have been inoculated with rhizobia, and more recently azospirillum, to increase their production or to allow them to be grown outside their original habitat. Application of Bacillus thuringiensis (BT) and other bacteria can help protect crops from insect infestation and plant diseases. With advances in genetic engineering, these bacteria have been manipulated for increased efficiency and expanded host range. Markers have also been added to aid in tracing the spread of the bacteria. The bacteria that naturally colonize certain crops have also been modified, in some cases to express the BT genes responsible for pest resistance. Pseudomonas strains of bacteria cause frost damage by nucleating water into ice crystals around themselves. This led to the development of ice- bacteria, which have the ice-forming genes removed. When applied to crops they can compete with the non-modified bacteria and confer some frost resistance. 108. This artwork is made with bacteria modified to express 8 different colors of fluorescent proteins. Other uses for genetically modified bacteria include bioremediation, where the bacteria are used to convert pollutants into a less toxic form. Genetic engineering can increase the levels of the enzymes used to degrade a toxin or to make the bacteria more stable under environmental conditions. 109. Bioart has also been created using genetically modified bacteria. In the 1980s, artist John Davis and geneticist Dana Boyd converted the Germanic symbol for femininity into binary code and then into a DNA sequence, which was then expressed in Escherichia coli. 110. This was taken a step further in 2012, when a whole book was encoded onto DNA. 111. Paintings have also been produced using bacteria transformed with fluorescent proteins. 110. Viruses. Viruses are often modified so they can be used as vectors for inserting genetic information into other organisms. This process is called transduction, and if successful, the recipient of the introduced DNA becomes a GMO. Different viruses have different efficiencies and capabilities. Researchers can use this to control for various factors, including the target location, insert size, and duration of gene expression. Any dangerous sequences inherent in the virus must be removed, while those that allow the gene to be delivered effectively are retained. 112. While viral vectors can be used to insert DNA into almost any organism, it is especially relevant for its potential in treating human disease. Although primarily still at trial stages, 113, there has been some successes using gene therapy to replace defective genes. This is most evident in curing patients with severe combined immunodeficiency rising from adenosine deaminase deficiency, ADA-SCID, 114, although the development of leukemia in some ADA-SCID patients, 115, along with the death of Jesse Gelsinger in a 1999 trial set back the development of this approach for many years. 116, in 2009, another breakthrough was achieved when an 8-year-old boy with Leber's congenital amaurosis regained normal eyesight, 116, and in 2016 GlaxoSmithKline gained approval to commercialize a gene therapy treatment for ADA-SCID. 114. As of 2018, there are a substantial number of clinical trials underway, including treatments for hemophilia, glioblastoma, chronic granulomatous disease, cystic fibrosis and various cancers. 115. The most common virus used for gene delivery comes from adenoviruses, as they can carry up to 7.5 kilobits of foreign DNA and infect a relatively broad range of host cells, although they have been known to elicit immune responses in the host and only provide short-term expression. Other common vectors are adeno-associated viruses, which have lower toxicity and longer-term expression, but can only carry about 4 kilobits of DNA. 115 Herpes simplex viruses make promising vectors, having a carrying capacity of over 30 kilobits and providing long-term expression, although they are less efficient at gene delivery than other vectors. 117 The best vectors for long-term integration of the gene into the host genome are retroviruses, but their propensity for random integration is problematic. Lentiviruses are a part of the same family as retroviruses, with the advantage of infecting both dividing and non-dividing cells, whereas retroviruses only target dividing cells. Other viruses that have been used as vectors include alphaviruses, flaviviruses, measles viruses, rhabdoviruses, Newcastle disease virus, pox viruses, and picornaviruses. 115. Most vaccines consist of viruses that have been attenuated, disabled, weakened or killed in some way so that their virulent properties are no longer effective. Genetic engineering could theoretically be used to create viruses with the virulent genes removed. This does not affect the virus's infectivity, invokes a natural immune response, and there is no chance that they will regain their virulence function, which can occur with some other vaccines. As such, they are generally considered safer and more efficient than conventional vaccines, although concerns remain over non-target infection, potential side effects, and horizontal gene transfer to other viruses. 118 Another potential approach is to use vectors to create novel vaccines for diseases that have no vaccines available or the vaccines that do not work effectively, such as AIDS, malaria, and tuberculosis. 119 The most effective vaccine against tuberculosis, the Bacillus Calmette-Guerin (BCG) vaccine, only provides partial protection. A modified vaccine expressing AM tuberculosis antigen is able to enhance BCG protection. 120 It has been shown to be safe to use at phase 2 trials, although not as effective as initially hoped. 121 Other vector-based vaccines have already been approved and many more are being developed. 119 Another potential use of genetically modified viruses is to alter them so they can directly treat diseases. This can be through expression of protective proteins or by directly targeting infected cells. In 2004, researchers reported that a genetically modified virus that exploits the selfish behavior of cancer cells might offer an alternative way of killing tumors. 123 Since then, several researchers have developed genetically modified oncolytic viruses that show promise as treatments for various types of cancer. 124, 125, 126, 127, 128 In 2017, researchers genetically modified a virus to express spinach defensin proteins. The virus was injected into orange trees to combat citrus greening disease that had reduced orange production by 70% since 2005. 129 Natural viral diseases, such as myxomatosis and rabbit hemorrhagic disease, have been used to help control pest populations. Over time, the surviving pests become resistant, leading researchers to look at alternative methods. Genetically modified viruses that make the target animals infertile through immunocontraception have been created in the laboratory, 130, as well as others that target the developmental stage of the animal. 131 There are concerns with using this approach regarding virus containment, 130, and cross-species infection. 132 Sometimes the same virus can be modified for contrasting purposes. Genetic modification of the myxoma virus has been proposed to conserve European wild rabbits in the Iberian Peninsula and to help regulate them in Australia. To protect the Iberian species from viral diseases, the myxoma virus was genetically modified to immunize the rabbits, while in Australia the same myxoma virus was genetically modified to lower fertility in the Australian rabbit population. 133 Outside of biology, scientists have used a genetically modified virus to construct a lithium-ion battery and other nanostructured materials. It is possible to engineer bacteriophages to express modified proteins on their surface and join them up in specific patterns, a technique called phage display. These structures have potential uses for energy storage and generation, biosensing and tissue regeneration with some new materials currently produced including quantum dots, liquid crystals, nanorings and nanofibers. 134 The battery was made by engineering M13 bacteriophages so they would coat themselves in iron phosphate and then assemble themselves along a carbon nanotube. This created a highly conductive medium for use in a cathode, allowing energy to be transferred quickly. They could be constructed at lower temperatures with non-toxic chemicals, making them more environmentally friendly. 135 Fungi. Fungi can be used for many of the same processes as bacteria. For industrial applications, yeasts combine the bacterial advantages of being a single-celled organism that is easy to manipulate and grow with the advanced protein modifications found in eukaryotes. They can be used to produce large complex molecules for use in food, pharmaceuticals, hormones, and steroids. 136 Yeast is important for wine production, and as of 2016 two genetically modified yeasts involved in the fermentation of wine have been commercialized in the United States and Canada. One has increased malolactic fermentation efficiency, while the other prevents the production of dangerous ethyl carbamate compounds during fermentation. 96 There have also been advances in the production of biofuel from genetically modified fungi. 137 Fungi, being the most common pathogens of insects, make attractive biopesticides. Unlike bacteria and viruses, they have the advantage of infecting the insects by contact alone, although they are outcompeted in efficiency by chemical pesticides. Genetic engineering can improve virulence, usually by adding more virulent proteins. 138 Increasing infection rate or enhancing spore persistence. 139 Many of the disease-carrying vectors are susceptible to entomopathogenic fungi. An attractive target for biological control are mosquitoes, vectors for a range of deadly diseases, including malaria, yellow fever and dengue fever. Mosquitoes can evolve quickly, so it becomes a balancing act of killing them before the plasmodium they carry becomes the infectious disease, but not so fast that they become resistant to the fungi. By genetically engineering fungi like Metarrhizum anisipli and Bovaria bassiana to delay the development of mosquito infectiousness, the selection pressure to evolve resistance is reduced. 140. Another strategy is to add proteins to the fungi that block transmission of malaria. 140. Or remove the plasmodium altogether. 141. Agaricus bisporus, the common white button mushroom, has been gene edited to resist browning, giving it a longer shelf life. The process used CRISPR to knock out a gene that encodes polyphenol oxidase. As it didn't introduce any foreign DNA into the organism, it was not deemed to be regulated under existing GMO frameworks and is such as the first CRISPR-edited organism to be approved for release. 142. This has intensified debates as to whether gene-edited organisms should be considered genetically modified organisms. 143. And how they should be regulated. 144. Plants. Tissue culture used to regenerate Arabidopsis thaliana. Plants have been engineered for scientific research, to display new flower colors, deliver vaccines, and to create enhanced crops. Many plants are pluripotent, meaning that a single cell from a mature plant can be harvested and under the right conditions can develop into a new plant. This ability can be taken advantage of by genetic engineers. By selecting for cells that have been successfully transformed in an adult plant, a new plant can then be grown that contains the transgene in every cell through a process known as tissue culture. 145. Much of the advances in the field of genetic engineering has come from experimentation with tobacco. Major advances in tissue culture and plant cellular mechanisms for a wide range of plants has originated from systems developed in tobacco. 146. It was the first plant to be altered using genetic engineering and is considered a model organism for not only genetic engineering, but a range of other fields. 147. As such, the transgenic tools and procedures are well established, making tobacco one of the easiest plants to transform. 148. Another major model organism relevant to genetic engineering is Arabidopsis thaliana. Its small genome and short life cycle makes it easy to manipulate, and it contains many homologs to important crop species. 149. It was the first plant sequenced, has a host of online resources available, and can be transformed by simply dipping a flower in a transformed agrobacterium solution. 150. In research, plants are engineered to help discover the functions of certain genes. The simplest way to do this is to remove the gene and see what phenotype develops compared to the wild-type form. Any differences are possibly the result of the missing gene. Unlike mutagenesis, genetic engineering allows targeted removal without disrupting other genes in the organism. 145. Some genes are only expressed in certain tissues, so reporter genes, like GUS, can be attached to the gene of interest, allowing visualization of the location. 151. Other ways to test a gene is to alter it slightly and then return it to the plant and see if it still has the same effect on phenotype. Other strategies include attaching the gene to a strong promoter and see what happens when it is overexpressed, forcing a gene to be expressed in a different location or at different developmental stages. 145. Suntory, blue, rose. Some genetically modified plants are purely ornamental. They are modified for flower color, fragrance, flower shape, and plant architecture. 152. The first genetically modified ornamentals commercialized altered color. 153. Carnations were released in 1997, with the most popular genetically modified organism, a blue rose, actually lavender or mauve, created in 2004. 154. The roses are sold in Japan, the United States, and Canada. 155. 156. Other genetically modified ornamentals include chrysanthemum and petunia. 152. As well as increasing aesthetic value, there are plans to develop ornamentals that use less water or are resistant to the cold, which would allow them to be grown outside their natural environments. 157. It has been proposed to genetically modify some plant species threatened by extinction to be resistant to invasive plants and diseases, such as the emerald ash borer in North American and the fungal disease, Ceratocystis platani, in European plane trees. 158. The papaya ring spot virus devastated papaya trees in Hawaii in the 20th century until transgenic papaya plants were given pathogen-derived resistance. 159. However, genetic modification for conservation in plants remains mainly speculative. A unique concern is that a transgenic species may no longer bear enough resemblance to the original species to truly claim that the original species is being conserved. Instead, the transgenic species may be genetically different enough to be considered a new species, thus diminishing the conservation worth of genetic modification. 158. Crops. Wild-type peanut top and transgenic peanut with Bacillus thuringiensis gene added, bottom, exposed to cornstalk borer larva. Genetically modified crops are genetically modified plants that are used in agriculture. The first crops developed were used for animal or human food and provide resistance to certain pests, diseases, environmental conditions, spoilage or chemical treatments, e.g. resistance to a herbicide. The second generation of crops aim to improve the quality, often by altering the nutrient profile. Third generation genetically modified crops could be used for non-food purposes, including the production of pharmaceutical agents, biofuels, and other industrially useful goods, as well as for bioremediation. 160. Kenyans examining insect-resistant transgenic Bacillus thuringiensis (BT) corn. There are three main aims to agricultural advancement: increased production, improved conditions for agricultural workers, and sustainability. GM crops contribute by improving harvests through reducing insect pressure, increasing nutrient value, and tolerating different abiotic stresses. Despite this potential, as of 2018, the commercialized crops are limited mostly to cash crops like cotton, soybean, maize, and canola, and the vast majority of the introduced traits provide either herbicide tolerance or insect resistance. 160. Soybeans accounted for half of all genetically modified crops planted in 2014. 161. Adoption by farmers has been rapid. Between 1996 and 2013, the total surface area of land cultivated with GM crops increased by a factor of 100. 162. Geographically, though the spread has been uneven, with strong growth in the Americas and parts of Asia and little in Europe and Africa. 160. Its socioeconomic spread has been more even, with approximately 54% of worldwide GM crops grown in developing countries in 2013. 162. Although doubts have been raised. 163. Most studies have found growing GM crops to be beneficial to farmers through decreased pesticide use as well as increased crop yield and farm profit. 164, 165, 166. The majority of GM crops have been modified to be resistant to selected herbicides, usually a glyphosate or glufosinate-based one. Genetically modified crops engineered to resist herbicides are now more available than conventionally bred resistant varieties. 167. In the USA, 93% of soybeans and most of the GM maize grown is glyphosate tolerant. 168. Most currently available genes used to engineer insect resistance come from the Bacillus thuringiensis bacterium and code for delta endotoxins. A few use the genes that encode for vegetative insecticidal proteins. 169. The only gene commercially used to provide insect protection that does not originate from B. thuringiensis is the cowpea trypsin inhibitor, CPTI. CPTI was first approved for use cotton in 1999 and is currently undergoing trials in rice. 170. 171. Less than 1% of GM crops contained other traits, which include providing virus resistance, delaying senescence and altering the plant's composition. 161. Golden rice compared to white rice. Golden rice is the most well-known GM crop that is aimed at increasing nutrient value. It has been engineered with 3 genes that biosynthesize beta-carotene, a precursor of vitamin A, in the edible parts of rice. 70. It is intended to produce a fortified food to be grown and consumed in areas with a shortage of dietary vitamin A. 172. A deficiency which each year is estimated to kill 670,000 children under the age of 5. 173. And cause an additional 500,000 cases of irreversible childhood blindness. 174. The original golden rice produced 1.6 micrograms per gram of the carotenoids, with further development increasing this 23 times. 175. It gained its first approvals for use as food in 2018. 176. Plants and plant cells have been genetically engineered for production of biopharmaceuticals in bioreactors, a process known as farming. Work has been done with duckweed lemna minor, 177, the algae Chlamydomonas reinhardti, 178, and the moss Physcometrella patens. 179, 180. Biopharmaceuticals produced include cytokines, hormones, antibodies, enzymes and vaccines, most of which are accumulated in the plant seeds. Many drugs also contain natural plant ingredients and the pathways that lead to their production have been genetically altered or transferred to other plant species to produce greater volume. 181. Other options for bioreactors are biopolymers, 182, and biofuels. 183. Unlike bacteria, plants can modify the proteins post-translationally, allowing them to make more complex molecules. They also pose less risk of being contaminated. 184. Therapeutics have been cultured in transgenic carrot and tobacco cells, 185, including a drug treatment for Gaucher's disease. 186. Vaccine production and storage has great potential in transgenic plants. Vaccines are expensive to produce, transport, and administer, so having a system that could produce them locally would allow greater access to poorer and developing areas. 181. As well as purifying vaccines expressed in plants, it is also possible to produce edible vaccines in plants. Edible vaccines stimulate the immune system when ingested to protect against certain diseases. Being stored in plants reduces the long-term cost as they can be disseminated without the need for cold storage, don't need to be purified, and have long-term stability. Also, being housed within plant cells provides some protection from the gut acids upon digestion. However, the cost of developing, regulating, and containing transgenic plants is high, leading to most current plant-based vaccine development being applied to veterinary medicine, where the controls are not as strict. 187. Genetically modified crops have been proposed as one of the ways to reduce farming-related CO2 emissions due to higher yield, reduced use of pesticides, reduced use of tractor fuel and no-tillage. According to a 2021 study, in EU alone widespread adoption of GE crops would reduce greenhouse gas emissions by 33 million tons of CO2 equivalent or 7.5% of total farming-related emissions. 188. Animals. The vast majority of genetically modified animals are at the research stage with the number close to entering the market remaining small. 189. As of 2018 only 3 genetically modified animals have been approved, all in the USA. A goat and a chicken have been engineered to produce medicines and a salmon has increased its own growth. 190. Despite the differences and difficulties in modifying them, the end aims are much the same as for plants. GM animals are created for research purposes, production of industrial or therapeutic products, agricultural uses, or improving their health. There is also a market for creating genetically modified pets. 191. Mammals. Some chimeras, like the blotched mouse shown, are created through genetic modification techniques like gene targeting. The process of genetically engineering mammals is slow, tedious, and expensive. However, new technologies are making genetic modifications easier and more precise. 192. The first transgenic mammals were produced by injecting viral DNA into embryos and then implanting the embryos in females. 60. The embryo would develop and it would be hoped that some of the genetic material would be incorporated into the reproductive cells. Then researchers would have to wait until the animal reached breeding age and then offspring would be screened for the presence of the gene in every cell. The development of the CRISPR-Cas9 gene editing system is a cheap and fast way of directly modifying germ cells, effectively having the amount of time needed to develop genetically modified mammals. 193. A porcine model of hemophilia A. Mammals are the best models for human disease, making genetic engineered ones vital to the discovery and development of cures and treatments for many serious diseases. Knocking out genes responsible for human genetic disorders allows researchers to study the mechanism of the disease and to test possible cures. Genetically modified mice have been the most common mammals used in biomedical research, as they are cheap and easy to manipulate. Pigs are also a good target as they have a similar body size and anatomical features, physiology, pathophysiological response and diet. 194 non-human primates are the most similar model organisms to humans, but there is less public acceptance towards using them as research animals. 195 In 2009, scientists announced that they had successfully transferred a gene into a primate species, marmosets, for the first time. 197 Their first research target for these marmosets was Parkinson's disease, but they were also considering amyotrophic lateral sclerosis and Huntington's disease. 198 Human proteins expressed in mammals are more likely to be similar to their natural counterparts than those expressed in plants or microorganisms. Stable expression has been accomplished in sheep, pigs, rats and other animals. In 2009, the first human biological drug produced from such an animal, a goat, was approved. The drug, Atrin, is an anticoagulant which reduces the probability of blood clots during surgery or childbirth and is extracted from the goat's milk. 199 Human alpha-1 antitrypsin is another protein that has been produced from goats and is used in treating humans with this deficiency. 200 Another medicinal area is in creating pigs with greater capacity for human organ transplants, xenotransplantation. Pigs have been genetically modified so that their organs can no longer carry retroviruses, 201, or have modifications to reduce the chance of rejection. 203 Chimeric pigs could carry fully human organs. 204 The first human transplant of a genetically modified pig heart occurred in 2023, 205, and kidney in 2024. 207 Livestock are modified with the intention of improving economically important traits such as growth rate, quality of meat, milk composition, disease resistance and survival. Animals have been engineered to grow faster, be healthier, 208, and resist diseases. 209 Modifications have also improved the wool production of sheep and udder health of cows. 189 Goats have been genetically engineered to produce milk with strong spiderweb-like silk proteins in their milk. 210 A GM pig called EnviroPig was created with the capability of digesting plant phosphorus more efficiently than conventional pigs. 212 They could reduce water pollution since they excrete 30 to 70% less phosphorus in manure. 213 Dairy cows have been genetically engineered to produce milk that would be the same as human breast milk. 214 This could potentially benefit mothers who cannot produce breast milk but want their children to have breast milk rather than formula. 216 Researchers have also developed a genetically engineered cow that produces allergy-free milk. 217 Mice expressing the green fluorescent protein. Scientists have genetically engineered several organisms, including some mammals, to include green fluorescent protein, GFP, for research purposes. 218 GFP and other similar reporting genes allow easy visualization and localization of the products of the genetic modification. 219 Fluorescent pigs have been bred to study human organ transplants, regenerating ocular photoreceptor cells, and other topics. 220 In 2011, green fluorescent cats were created to help find therapies for HIV, AIDS and other diseases. 221 As feline immunodeficiency virus is related to HIV. 222 There have been suggestions that genetic engineering could be used to bring animals back from extinction. It involves changing the genome of a close living relative to resemble the extinct one and is currently being attempted with the passenger pigeon. 223 Genes associated with the woolly mammoth have been added to the genome of an African elephant, although the lead researcher says he has no intention of creating live elephants and transferring all the genes and reversing years of genetic evolution is a long way from being feasible. 225 It is more likely that scientists could use this technology to conserve endangered animals by bringing back lost diversity or transferring evolved genetic advantages from adapted organisms to those that are struggling. 226 Humans. Gene therapy. 227 Uses genetically modified viruses to deliver genes which can cure disease in humans. Although gene therapy is still relatively new, it has had some successes. It has been used to treat genetic disorders such as severe combined immunodeficiency, 228, and Leber's congenital amaurosis. 229 Treatments are also being developed for a range of other currently incurable diseases, such as cystic fibrosis, 230 sickle cell anemia, 231 Parkinson's disease, 232 233 cancer, 234 235 236 diabetes, 237 heart disease, 238 and muscular dystrophy. 239 These treatments only affect somatic cells, meaning any changes would not be inheritable. Germline gene therapy results in any change being inheritable, which has raised concerns within the scientific community. 241 In 2015, CRISPR was used to edit the DNA of non-viable human embryos. 243 In November 2018, He Jiankui announced that he had edited the genomes of 2 human embryos, in an attempt to disable the CCR5 gene, which codes for a receptor that HIV uses to enter cells. He said that twin girls, Lulu and Nana, had been born a few weeks earlier and that they carried functional copies of CCR5 along with disabled CCR5 mosaicism and were still vulnerable to HIV. The work was widely condemned as unethical, dangerous, and premature. 244 Fish. When exposed to 13 degrees Celsius water, the zebrafish modified to express a carp creatine kinase right maintained swimming behavior, while wild-type zebrafish left could not. 200 He said that twin girl In 2015, CRISPR was used to edit the DNA of non-viable human embryos. 243. In November 2018, He Jiankui announced that he had edited the genomes of 2 human embryos, in an attempt to disable the CCR5 gene, which codes for a receptor that HIV uses to enter cells. He said that twin girls, Lulu and Nana, had been born a few weeks earlier and that they carried functional copies of CCR5 along with disabled CCR5 mosaicism and were still vulnerable to HIV. The work was widely condemned as unethical, dangerous, and premature. 244. Fish. When exposed to 13 degrees Celsius water, the zebrafish modified to express a carp creatine kinase right maintained swimming behavior, while wild-type zebrafish left could not. 245. Genetically modified fish are used for scientific research, as pets and as a food source. Aquaculture is a growing industry, currently providing over half the consumed fish worldwide. 246. Through genetic engineering it is possible to increase growth rates, reduce food intake, remove allergenic properties, increase cold tolerance and provide disease resistance. Fish can also be used to detect aquatic pollution or function as bioreactors. 247. Several groups have been developing zebrafish to detect pollution by attaching fluorescent proteins to genes activated by the presence of pollutants. The fish will then glow and can be used as environmental sensors. 248. 249. The glowfish is a brand of genetically modified fluorescent zebrafish with bright red, green, and orange fluorescent color. It was originally developed by one of the groups to detect pollution, but is now part of the ornamental fish trade, becoming the first genetically modified animal to become publicly available as a pet when in 2003 it was introduced for sale in the USA. 250. GM fish are widely used in basic research in genetics and development. 2 species of fish, zebrafish and medaka, are most commonly modified because they have optically clear chorions, membranes in the egg rapidly develop, and the one-cell embryo is easy to see and microinject with transgenic DNA. 251. Zebrafish are model organisms for developmental processes, regeneration, genetics, behavior, disease mechanisms and toxicity testing. 252. Their transparency allows researchers to observe developmental stages, intestinal functions and tumor growth. 253. 254. The generation of transgenic protocols, whole organism, cell or tissue specific, tagged with reporter genes, has increased the level of information gained by studying these fish. 255. GM fish have been developed with promoters driving an overproduction of growth hormone for use in the aquaculture industry to increase the speed of development and potentially reduce fishing pressure on wild stocks. This has resulted in dramatic growth enhancement in several species, including salmon, 256 trout, 257, and tilapia. 258. Aquabounty Technologies, a biotechnology company, have produced a salmon called Aqua Advantage Salmon that can mature in half the time as wild salmon. 259. It obtained regulatory approval in 2015, the first non-plant GMO food to be commercialized. 260. As of August 2017, GMO salmon is being sold in Canada. 261. Sales in the U.S. started in May 2021. 262. Insects. Overexpression of methyl CpG binding protein 2 in Drosophila impairs climbing ability, right compared to the control group, left. 263. In biological research, transgenic fruit flies, Drosophila melanogaster, are model organisms used to study the effects of genetic changes on development. 264. Fruit flies are often preferred over other animals due to their short life cycle and low maintenance requirements. They also have a relatively simple genome compared to many vertebrates, with typically only one copy of each gene, making phenotypic analysis easy. 265. Drosophila have been used to study genetics and inheritance, embryonic development, learning, behavior, and aging. 266. The discovery of transposons, in particular the P element, in Drosophila provided an early method to add transgenes to their genome, although this has been taken over by more modern gene editing techniques. 267. Due to their significance to human health, scientists are looking at ways to control mosquitoes through genetic engineering. Malaria-resistant mosquitoes have been developed in the laboratory by inserting a gene that reduces the development of the malaria parasite, 268, and then use homing endonucleases to rapidly spread that gene throughout the male population, known as a gene drive. 269. 270. This approach has been taken further by using the gene drive to spread a lethal gene. 271. 272. In trials, the populations of Aedes aegypti mosquitoes, the single most important carrier of dengue fever and Zika virus, were reduced by between 80% and by 90%. 273. 274. 272. Another approach is to use a sterile insect technique, whereby males genetically engineered to be sterile outcompete viable males, to reduce population numbers. 275. Other insect pests that make attractive targets are moths. Diamondback moths cause United States dollars to $5 billion of damage each year worldwide. 276. The approach is similar to the sterile technique tested on mosquitoes, where males are transformed with a gene that prevents any females born from reaching maturity. 277. They underwent field trials in 2017. 276. Genetically modified moths have previously been released in field trials. 278. In this case, a strain of pink bollworm that were sterilized with radiation were genetically engineered to express a red fluorescent protein, making it easier for researchers to monitor them. 279. Silkworm, the larva stage of Bombyx mori, is an economically important insect in sericulture. Scientists are developing strategies to enhance silk quality and quantity. There is also potential to use the silk-producing machinery to make other valuable proteins. 280. Proteins currently developed to be expressed by silkworms include human serum albumin, human collagen alpha chain, mouse monoclonal antibody and N-glycanase. 281. Silkworms have been created that produce spider silk, a stronger but extremely difficult to harvest silk. 282. And even novel silks. 283. Other. Frog expressing green fluorescent protein. Systems have been developed to create transgenic organisms in a wide variety of other animals. Chickens have been genetically